E-level biology 9700 paper 4 variant 2 February March 2026, and this is the fourth video on this paper in which we are going to discuss question number 7 and 8. So the question number 7 is the process of ultrafiltration occurs in the Bowman's capsule region of the kidney nephron. Figure 7.1 is a diagram representing part of a Bowman's capsule in which they have labeled three parts, and they have shown the lumen of Bowman's capsule and the lumen of capillary glomerulus. So we are going with these three parts. The part A is, you can see this is a just like a foot-like process pedicels that wrap around the glomerular capillary and form filtration slits. So this is the podocyte. This is the podocyte, and B is the you can see this is the basement membrane, which acts as the main filtration barrier, preventing large proteins from passing into the filtrate. So this is the basement membrane. And C is the, you can see the endothelial line cell. So they have labeled only one, but actually this is the endothelial lining. So these cells contain pores, fenestrations that allow water and small to pass through the solutes to pass through the while remaining food cells. So this is the endothelial cell. So now we are moving towards the next question, and next question is A was podocyte, B was basement membrane, and third was endothelial cell. Podocyte, basement membrane, endothelial cell, I have not written on the given space, but this is all about them. So the kidney function of a person can be assessed by measuring the glomerular filtration rate, GFR. The GFR is the rate at which fluid filters from the blood in the glomerulus into the Bowman's capsule, suggest factors that are likely to decrease the GFR. So we have to give the suggestion about the factors that will decrease the glomerular filtration rate. So first of all, the decreased blood pressure or low blood pressure can be the cause of it. Secondly, the constriction of afferent arteriole, uh, that can be the reason. And thirdly, the dilation of the afferent uh, arteriole or blockage in the urinary tract. So the damage or disease of the filtration barrier, so increased plasma solute concentration, higher blood oncotic pressure, all they can be the causes. So we have to go with them as first of all, the decreased blood pressure or low blood pressure due to severe dehydration, blood loss, or any other. For example, due to severe dehydration, blood loss, etc., or shock. So it can be the reason. Secondly, the constriction of the afferent arteriole, which can be due to the sympathetic nervous stimulation. due to sympathetic nervous stimulation. And thirdly, the blockage in the urinary tract due to kidney stones, enlarged prostate, or damage or disease of the filtration barrier, or increased, I can write them as damage of the, you can choose any three of them, or increased plasma solute concentration. It was part B. Now moving towards the third part, which is C. The kidney has an important role in homeostasis. So explain what is meant by the term homeostasis and describe the principles of homeostasis in mammals. So here we have to write first of all uh, we have to define the homeostasis and then the different principles due to which the homeostasis takes place in the body. So the homeostasis is the maintenance of the relatively constant internal environment. For example, the different concentrations at blood glucose concentration, water potential, core body temperature, and within narrow limits of pH, all they should be. So the principles of homeostasis is actually the negative feedback mechanism by which the homeostasis is kept inside the body. So there are receptor cells which detect any change or deviation of a physiological parameter from a set point, and then the control center, and then effectors, and then dynamic equilibrium is kept inside, and then coordination system is also involved into it with nervous system and endocrine system. So its importance is that it prevents damage to cells enzymes by maintaining optimal conditions, so preventing enzyme denaturation. So all this takes place when the body is under control of this homeostasis. So I'm going to start the question right here. As first of all, the homeostasis is the maintenance of a relatively constant internal environment. Blood glucose, blood pressure, water potential, etc. So the principles of the principle negative feedback mechanism. Detect receptors, components of this homeostasis. Detect receptors, detect any change in the environment, controls message is sent to control center, which is brain or hypothalamus, brain or to receive the incoming sensory information. Effectors then respond or to reverse or counteract the initial change. Bringing back to set point. Two key dynamic equilibrium coordination systems relay relies on the nervous system. Nervous system, electrical chemical signals along with neurons and the endocrine system. And the endocrine system consists of hormones and to transport the signals between receptors and effectors. So the importance of this importance of this mechanism is that it prevents damage to cells by maintaining optimal conditions. Cells or enzymes by maintaining in optimal conditions. So it was all about question number 7. Now moving towards the question number 8. And question number 8 is, the figure shows uh, three processes that require energy in living organisms. As for example, you can see the A, in which the lower concentration of substance, and they show the movement towards the through a membrane towards the high concentration of substance. It means this is active transport. And here you can see in the B, the monomers, they are joining together to make the uh, polymers, or you can see that the anabolism or condensation reaction is taking place over here. And C is, you can see the light and chlorophyll. Chlorophyll is capturing the light, and then you can see this is the photoactivation reaction, which is light dependent reaction, absorption of light by chlorophyll resulting in the excitation of the electrons. So these are the three which can be written over here. A is the active transport, in which energy is required when the molecules move from their lower concentration towards the high concentration, so they use energy. And B is the, you can see over here, B was the anabolism, concentration, or polymerization, any one of them you can write. Or polymerization. You can write any one of them. C is photoactivation. Now there is B part. ATP produced in mitochondria by a process called chemiosmosis. So outline the process of chemiosmosis in mitochondria. So here you can see, first of all, the protein pumping takes place as high energy electrons pass along the electron transport chain located on the inner mitochondrial membrane. So released in using the hydrogen pumps from the, and then this establishes a high concentration gradient of protons and an electrochemical gradient across the inner membrane. Then diffusion through ATP synthase, protons diffuse down their concentration, their electrochemical gradient back into the matrix through ATP synthase. So channels, so the ATP synthesis, the flow of protons through ATP synthase provides the energy to photophosphorylase and inorganic phosphate to form. So it was little bit in detail, but these are the four outlines. So we have to outline them as first of all the pass through or along the electron transport chain. I think it should be along the on the inner mitochondrial membrane. Inner mitochondrial membrane. Released energy is used to form hydrogen. Then proton grade, this establishes a high concentration gradient protons diffuse down it is little bit in detail for your convenience to for your understanding as well. You can make outlines from this. Across the inner membrane. Then the flow of protons through ATP synthesis. The flow of protons through ATP synthesis. Synthesis provides the energy to phosphorylate and inorganic phosphate to and inorganic phosphate to form. So you can make outlines from this. Now the last part of this question is so different respiratory substrates have different energy values. Table 8.1 shows the approximate energy values of carbohydrate and lipids. So the table 8.1 respiratory substrate approximately energy values are carbohydrate and lipid. So students here, they have given us the value of carbohydrates in 17 kilojoules per gram and lipid 38.100. So calculate the mass of carbohydrates needed to release the same quantity of energy as 100 gram of lipid. So there they have given uh, the the lipid producing 37.8. So first of all, we have to calculate the total energy from 100 gram of lipid. So I will make use of the whole space over here. First part is to calculate total energy from 100 gram of lipid. So this is the total energy is equal to into 37.8. So it will be equal to, you can make a calculation as, you can multiply 37.8. This is very simple, you can say. So it will give rise the answer 3780 kilojoule. And second calculation is also very simple, which is calculate mass of carbohydrates needed to supply 3780 kilojoule. So mass of carbohydrate is equal to 3780 divided by 17.0. So the answer will be, if you calculate, 222.4 gram. So it can be the answer if you calculate it by yourself. So it was all about question number 7 and 8. Please like, share, and subscribe my channel to watch more videos on these papers and the coming onset of this May June session. So thanks for watching.